Hello, everyone, and welcome to today's live broadcast, Understanding Cancer, Cell Matrix Interactions and the 3D Cell Migration, presented by Nicholas Kurniawan, PhD, Assistant Professor in the Department of Biomedical Engineering at Eindhoven University of Technology. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Bitplane. Bitplane is the world's leading interactive microscopy image analysis software company known for Imaris, which is actively shaping the way microscopic images are processed through constant innovation and a clear focus on 3D and 4D imaging, analysis, and data exploration. To learn more about Bitplane, please visit www.bitplane.com. Today's webinar is a part of the Search series where Bitplane invites researchers from the top scientific and medical institutions who have dedicated their work to discover new therapeutic agents or vaccines for some of the most severe diseases of the 21st century. They are delighted to discover that in many cases, Imaris has pushed the research forward, putting the scientists closer to finding new therapies. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. This presentation has been approved for continuing educational credits. If you want to obtain the credit, please click on the Get Your Free CME CE Credits button located in the lower left of your screen. It will take you through the process to obtain your certification. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Nicholas Kurniawan. He received his PhD in 2012 from the National University of Singapore, studying the role of matrix viscoelasticity in cancer metastasis. He then carried out his postdoctoral research as a Marie Curie Fellow in Foam Institute, Almhof, Amsterdam, in the Netherlands, investigating the hierarchical structure property relation in the cytoskeleton and extracellular matrices. His works have provided important insights into the underlying physical mechanisms behind the intriguing nonlinear behaviors of protein networks. In 2015, he joined Eindhoven University of Technology in Eindhoven, the Netherlands, as an assistant professor in the biomedical engineering department. Combining his expertise on biopolymer physics, cell biomechanics, and cytoskeletal rheology, his current research focuses on the biophysical and mechanobiological aspects of cell matrix interactions and the applications in regenerative medicine. Please join me in welcoming Nicholas Kurniawan. I will now turn the presentation over to him.
All right, thanks, Judy, for the introduction. It's a pleasure to be here and speaking to you guys today. And for that, I'll thank, first of all, the people from Bitplane and LabRoots for making this event happen and for bringing us together today for this presentation. So, I'm going to talk about cell matrix interactions today, and in particular, how the interactions between the cells and the matrix affect the way that cells migrate in three-dimensional environment, especially in the context of cancer. So the primary research interest in my group is biology in physical context. So biology in context, in particular in the physical context of cell behavior. So basically, the primary questions that we are trying to answer is how are biological functions at the cell and tissue levels affected by the material properties of the cell and the tissues. So I think in this particular case, cancer is a particularly good example, a prime example of how cell matrix interactions can really affect or even influence, dramatically influence how cell behaves. So, well, yeah, probably you all know about cancer. I think, yeah, everyone knows about cancer, and this has been sort of depicted very, very nicely by Siddhartha Mukherjee in his Pulitzer Prize-winning book. So in 2010, so the book was written before 2010. So in 2010, about 600,000 Americans and more than 7 million humans around the world will die of cancer. In the United States, 1 in 3 women and 1 in 2 men will develop cancer during their lifetime. A quarter of all American deaths and about 15% of all deaths worldwide will be attributed to cancer. In some nations, cancer will surpass heart disease to become the most common cause of death. So it's obvious that, cancer is a big business, is a big threat to humankind. So what are really the characteristics of cancer that, makes it so devastating? So in 2 seminal papers by Hanahan and Weinberg, they sort of summarized the 6 or even 8 hallmarks of cancer. So it's shown in this slide. So basically, the cells can sustain proliferative signaling, meaning that they can basically divide and continue to divide much more than normal cells do. They can resist cell death, evade growth suppressors, and so on and so forth. And but what I think is most important is actually this right here, activating invasion and metastasis. So in fact, metastasis is responsible for 90% of solid tumor-related deaths. So what that means is, if you or somebody or a patient has cancer or tumor, but the tumor is isolated, is localized, and is not invading any other organ or tissue, then chances are you or the patient will be fine. We can remove the tissue containing the tumor, and then there will be no other or no further proliferation of cancer cells. But once the cancer cells have invaded other tissues and metastasized, then he's in a big trouble. So what is really happening during metastasis? So what's happening is you have this primary tumor. Some cells are able to get out of this primary tumor, migrate to the tissue, get into blood flow or bloodstream, and then circulate throughout our body, find a greener pasture somewhere, migrate again, and start a new colony. So there's a huge multi-stage process going on, and the cells have to migrate through a very, very different or diverse kind of tissue. So they have to migrate over here. So for example, this could be a breast tissue and get into the bloodstream, move together in the blood circulation, and migrate again through a very, very potentially very different kind of tissue, and then start a new colony. So really, the cells are subjected or they're exposed to many different kind of tissues with very different or diverse microstructure, and they need to be able to migrate through it and survive and also proliferate. And this is particularly interesting because there are many ways that the cells can migrate. So I'm showing 3 examples here. So the center example is basically how cells migrate in a wound healing assay. The right example is how cells are able to collectively migrate or move as a colony in what is termed collective cell migration. What is happening on the left video is individual cell migrating or moving following chemoattractants. So this pipette kind of thing is actually providing chemoattraction for the cell. the cell is able to sense that and migrate towards the source of the chemoattractant. And so coming back to the metastasis process, so I said before that in metastasis the cells have to migrate through potentially very different tissues with very diverse microstructure. So if you can imagine, if you have a cell migrating through breast tissue, and then it has to go to it finds a greener pasture in a bone and tries to migrate in a bone, the cell will experience or feel or sense very different environment or substrate. And this has been studied very elegantly in this paper by Lu et al. In 2000. So what they do is they plate cells on a substrate, this 2D substrate, and so half of the substrate is soft and the other half of the substrate is stiff. And apparently, they sort of monitored where the cells how the cells migrate and where the cells migrate. And apparently, the cells that started out on the soft side of the substrate, for example, this cell, they can hit the interface of the soft stiff interface, substrate interface, and happily move over to the stiff substrate. Whereas if the cells start from the stiff substrate, stiff half of the substrate, they hit the interface and they basically decide not to go into it. So there seems to be a preferential stiffness of the substrate for the cells. And yeah, apparently the cells behave differently. The morphology of the cell is different in soft and stiff substrates. And coming back to the idea of metastasis, different tissues in our body apparently have a very diverse mechanical properties. So, for example, brain has elasticity of on the order of 0.1 to 1 kilopascal, but muscles, for example, has about 10 to 100 times stiffer mechanical properties, about 10 kilopascals. Bones, obviously, is a lot more stiff. So cells really have to migrate through very different substrates with very different mechanical properties, and people start to look at how cells are able to achieve that, how cells are able to migrate through all these different tissues with different mechanical properties. So the two experimental assays that people have used so far in studying 3D cell migration is so either they put cells randomly in a matrix, basically just spread the cells everywhere in the matrix and let them sort of migrate in a random manner and then monitor the migration properties of these cells. Or they create spheroid of cells, basically a colony of cells, and they look at how cells are able to invade the matrix, the surrounding matrix, from this colony of cells. So these are, very simple in vitro assay. It provides great statistics. You can monitor hundreds of cells at one experiment, one assay, but varying matrix properties is typically not straightforward because the cells are in the matrix from the very beginning. So our idea was something like this, where you have cells in what we call inner gel. So this is the inner gel. We use collagen gel, so in our collagen gel. And we sort of encapsulate this inner gel in an outer gel without any cell. So the idea is we will let the cells migrate out from the inner gel to the outer gel and monitor how the cells are migrating through the outer gel. And if you're interested in the details, the experimental details, feel free to look at this paper where we describe in a video protocol how the assay is set up. So this is also simple. It mimics tumor topology. It provides great statistics, and importantly, varying matrix properties is straightforward because all you need to do is basically change the properties of the outer gel without doing anything to the inner gel. So the experimental model is something like this. You have a primary site at a center in the inner gel, and then we let the cells migrate out into the outer gel. And then we look at how the cells migrate through the outer gel. And this is where our experience with Imaris, the software Imaris provided by Bitplane, has been very good. So Imaris is able to track many cells simultaneously, and not only in 3D but also in 4D, so 3D plus time. So I mentioned the ability to change very easily the properties of the matrix and study how cells are able to migrate in different kind of matrix. So we did that, and what I'm plotting here, for example, is the speed of the cell migration and the histogram of the speed of the cell migration in different matrices. So for example, these are basically collagen gels of different densities, 1.5 to 1.5 mg per mil, and these are collagen gels with the same density but with different pH, polymerization pH. I will get back to this, why we do this kind of variations in a couple of slides. But that's not only the only thing that we get out. So the readout is the cell trajectory, and we can quantify, for example, cell speed, but also what we call persistence. So how persistent the cell is in the migration trajectory. And so one of the findings that we found was very interesting is this. So if you actually think of migrating cells, cells migrating in a population, and you think of it as a migrating wave, a wave of migrating population, you can sort of categorize the cells within the population as the cells right in front of the population, right, the migration front basically, or the cells behind them, the cells that are what we call in the migration region. And what we found was very interesting is apparently the speed of the cells in the migration front is higher, significantly higher than the speed of the cells behind them. So we are seeing in the gray bars here is the histogram of the cells at the migration front, and which is significantly higher than the cells behind them, the black bars. But not only that, but apparently the directionality is also very different. So if you quantify the persistence of the trajectory, apparently the cells at the front move with a lot more directionality. So in this example, I'm plotting a result, a sample where I'm looking at this particular spot in the sample. So the cells are predominantly moving to the left, whereas the cells behind them are migrating sort of more randomly. So at the same time, we are also looking at what is happening to the matrix, what is happening to the matrix surrounding the cells, the fiber network surrounding the cells. And obviously people have noted previously that the cells are able to do many things to the fibers surrounding them. So the cells can pull on the fibers, they can push the fibers away, they can, for example, degrade the matrix, degrade the fibers if they are sort of trapped in a very dense matrix, local matrix. They can deposit matrix. So there are many remodeling by the cell on the matrix, both physically and chemically. So having this in mind, we hypothesized, or we sort of speculated that what we were seeing previously was kind of a terrain effect. So what I mean by that is the cells at the migration front, what they have in front of them is really a very nice porous, matrix or fiber networks which are pristine, untouched by other cells. They move happily through it, and they migrate very quickly and with a lot of directionality. Whereas the cells behind them, what they have in front of them, is really something like this, a forest that has been cut down by cells behind, in front of them, with a lot of mechanical and biochemical cues around them, and basically the cells get confused. There are too many cues everywhere, and that sort of impedes the directionality of the cells, and that's why the cells move slower and with a lot more randomness in the migration region. So that's not the only thing that we did. So we started to mess with the wheel. So we started to mess with the molecular mechanisms that allow for cell migration. So for example, we added drugs that affect, for example, myosin activity, or affectin polymerization, or microtubule stability, or even drug that inhibit the ability of the cells to degrade a matrix, so MMP inhibitor. And so this is the result in terms of what is the change in cell speed. So delta S is the change in cell speed, and delta K is the change in cell persistence. And in most of the cases, the drugs produce a negative value for the speed, meaning that the cells are less able to migrate with this. The cells migrate with much reduced speed, but the extent of the efficacy of these drugs are different. So in this particular example, cytokinin D, which affects the polymerization of actin, is a lot more effective in terms of reducing cell speed, but not in terms of reducing cell persistence. But that's not the only thing. So we also started to do all these drug studies in different matrices. So all these bar graphs are actually showing for example, the change of cell speed and change of cell persistence in the different matrices, 2.5 mg per mil, 1.5, 4. So towards the right is basically increasing collagen density, and towards the bottom is increasing pore size. So basically, the pH, the polymerization pH of the collagen gel influences the pore size of the gel without affecting without changing the collagen density. So pH 9 produced gels with very small pores on the order of 1 microns, and if you go down to pH of 6, then you end up with a gel of, for example, I think it's about 5 microns, even with the same collagen density. So now you can start to scour this all this information from these bar graphs, and we certainly have spent many hours trying to analyze this and understand what is going on. But the main message that I'm going to discuss today in this presentation is that all the bars look different, and when all the bars look different, that means that some drugs are more effective than other drugs in some ECM, some matrix, and some drugs are more effective than in one matrix than in other matrices. So that means that there is no sort of universal drug, no best drug, and there's no universal matrix, and that the cells adapt. So that means that if you want to come up with a drug that impedes or inhibits cancer cell migrations, then you need to be able to test the efficacy of this drug in the proper context where the drug is going to be applied. So if, for example, you want to look at the efficacy of this drug the potency of this drug to, for example, inhibit cancer cell migrations in very dense matrices, then you need to test this in very dense matrices and not very porous matrix, and vice versa, of course. So what have we learned so far? So we've learned that, so we've came up with a very simple yet powerful in vitro study, in vitro experimental setup to study 3D cell migration quantitatively. We've known that individual cell migration is strongly affected by biophysical and also the structural properties of the local microenvironment, not just the bulk environment. And mechanistic plasticity allows cells to actively adapt, and this is a keyword. The cells can adapt their invasion machinery depending on the matrix, what they're having surrounding them. So all of these are, pointing us towards the matrix. So it is important to look at what happens at the matrix and how the cells are able to interact with the matrix, both physically and chemically. So people have been trying to understand cell behavior and how they respond to, for example, biochemical and mechanical stimuli, but they sort of think of it as a black box. But in reality, the cells are also able to change the environment. They can also send back biochemical and mechanical stimuli to the environment, to the matrix, which can then be sensed by other cells. So in essence, what I'm saying is that no man is an island. No cell is an island. So cells are never alone in the body, but most studies were done on isolated cells. So there's kind of a mismatch here, conceptual mismatch. And the environment and the interactions between the cell and the environment need to be considered. So I think, coming back to my first slide. So we are interested in biology in the physical context, and I think this is very, very beautifully captured in the two videos that I'm showing here. So these are videos captured by the group of Eric Bedzik. So what I'm showing on the left video is basically a cell, well, a couple of cells migrating, and what you're seeing in the cells, the colorful filaments in the cells, those are the cytoskeleton. So the cytoskeletons are very dynamic and remodeling all the time as the cells are actively migrating. But that's not the only picture, the only issue or thing that we need to consider. But outside of the cell, you also have this fibrous network. So this is just the green thing is a cell migrating within a collagen matrix. So outside of the cell, there's also these protein networks, and these protein networks are dynamically remodeled by the cells. So the question then is, what does the cell feel? If they're in this kind of protein networks, what do they feel? How can we know what they sense and how they interact with this environment? So, so I guess the most straightforward answer, way to answer this question is to try to feel what the cells feel. And people have been trying to characterize mechanically the environment, the matrix, and also protein networks and biopolymer networks using a bulk measurement like Microrheology. So what happens is you have a plate and another plate or a cone, and you put the samples in between, and you apply kind of a twisting motion or a shearing motion, and you look at the stress and strain, and you deduce what is the mechanical properties of the matrix. But that is a bulk measurement, and that is probably not what the cells are feeling. So we need another approach which can measure local properties, properties that the cells can sense for themselves. And one way to do that is using what is called microrheology. So what is happening in microrheology is, instead of measuring the entire sample in bulk, you put a probe inside the sample or the matrix. So in this case, it's just a microsphere or a bead, and you put them in, put these beads in the matrix, and while looking at the particle dynamics, how the beads move due to Brownian motion in this matrix or the sample, you can deduce things like microstructure, the microstructure of the matrix, and the local mechanical compliance, because what the probe is probing, what the bead is probing in this particular case, is just a very local environment, a very local matrix. It's not the entire sample. So this way you can really measure the mechanical properties at local scale, micro scale. So this is not a new idea. People have been doing this for several years, and people have in fact put beads or probes in the cell, inside of the cells, and looking at the particle dynamics of the probes in the cells, they can look at they can sort of measure the local viscoelasticity within the cells, within the cytoplasm. But what we are trying to do is look at measure what the cell is feeling. So basically, if this is a cell, we want to know what the cell is feeling over here. We want to know how stiff this particular region is, and so on and so forth. So that's basically what we did. So we put beads in the matrix surrounding the cells, and looking at the Brownian motion of these beads, we can calculate, for example, the elastic moduli and the loss modulus of the local matrix. So in this particular example, I'm showing you this is a cell, and this is the leading edge of the cell, and those small dots are basically our probes. So these are our probes. And so these are in front of the leading edge of the cell, and we also put probes around the cell body. So for example, these. And based on the motions of these beads, we can calculate the elastic modulus and the loss modulus. And what we are seeing is apparently the region in front of the cell has significantly higher loss modulus compared to the so this is in front of the leading edge of the cell, and this is around cell body. And basically, the elastic modulus, or the stiffness of the local matrix, doubles when the cells are sort of doing something on the matrix. So by looking at the same time at what the matrix is doing, we are able to see that in front of the matrix, in front of the leading edge of the matrix, there's a lot of bundling of the fibers, so in this case collagen fibers. So what basically that means is that the cells are able to grab onto collagen fibers and pull these fibers, just because of cell contractility, and pull these fibers towards them, and thereby creating bundles, thick bundles of collagen fibers. And that is what is causing this increase in stiffness locally in front of the matrix. So basically, cells can exert contractile forces to establish themselves, and these forces change the local properties of the matrix. These forces make the local matrix stiffer. And well, so you can think of it this way. So if you put a cell in a matrix, they do this kind of contractility, and they create this kind of small pockets of higher local stiffnesses. So you can hypothesize that if you put more and more cells, there will be a lot more of these pockets of high stiffness. So the hypothesis then is, if you put more and more cells, the tissue, the entire tissue or matrix, will become stiffer. And that is something that we can test, and apparently that is indeed the case. So if you put more and more cells, the stiffness of the matrix goes up significantly. But if you, for example, inhibit contractility of the cell by adding drugs or whatever, then you don't see that. So these asterisk symbols are the control measurements where contractility is inhibited. So that means that this accumulation of these local stiffenings due to the cell lead to large-scale, macro-scale tissue stiffening. But that's actually not the entire story. So I've been telling you what happens in, for example, collagen shells or other biopolymer shells like fibrin shells. But so people started to do this in different kind of matrices. And in this particular study, for example, they put cells in packed hydrogels, so packed-based hydrogels with RGD modified packs, which are with some ability. So they put RGD peptides on the pack and also peptides that are able to be degraded by the matrix, so by the cells. So when they put cells in there, and the pore size of this kind of packed hydrogels is very, very tiny, in the order of nanometers. The cells are basically initially stuck in the matrix, but they're able to degrade the surrounding matrix, and thereby they are able to move around. And this kind of experiment shows that when they did microrheology around the cells, the area, the local region surrounding the cells are actually more liquidy, more viscous than it is elastic. And this, especially the pericellular area, they become a lot more liquidy. Whereas the area, far from the cells, they are still kind of elastic solids. So what this study and our study show is that depending on the kind of matrix that the cells find themselves in, they can do different kind of modification or remodeling of the local matrix around them. So yeah, with that I'll just conclude our presentation today. And yeah, so the conclusion is actually, context, context, context. Context is super important if you're trying to understand the cell behavior in physiologically relevant situations. And of course, by context, what I mean is cell matrix interactions. So we need to be able to understand what kind of interactions the cell are able to do with the matrix, and how these interactions are translated and manifested in terms of the local mechanical properties as well as structural properties. But what I've described in this presentation is just, sort of scratching the surface. So I've described a little bit about the physical and mechanical aspect of cell matrix interactions. But in reality, in our body, for example, the interaction between the cell and the matrix takes place in a very, very complex way, involving many, many different factors, both physical or mechanical and biochemical. And all of these factors are, they are affecting each other in ways that we are still trying to understand. So all these arrows show you what people have shown to be, sort of factors that are affecting each other in terms of cell matrix interactions. And so the cell matrix interaction itself is also very dynamic. It's changing as a function of time, and so the matrix can affect cell behavior. At the same time, the cells can affect or can change the matrix properties, and this feedback loop between the cell and the matrix are still, this kind of feedback loops need still to be investigated further. And this is particularly important in the case of tumor. And of course, I haven't really delved much into tumor, microenvironment. But in tumor microenvironment, there's a lot of interactions not only between the cell and the matrix, but also between different kind of cells, different cell types. And I think what will be very beneficial in the future, and so this is what I envision will be the way forward in terms of understanding cell behavior, but also in terms of cancer, understanding cancer invasion and cancer progression, and also, trying to develop therapeutical interventions in cancer, but also in things like tissue engineering, where the interaction, the physical and mechanical and biochemical interaction between the cell and the matrix are very important in terms of, for example, designing scaffolds with desired properties to direct cell behavior. So what I think will be very important in the future is how we can integrate all this information. So, so the cell phenotype is basically going to be a function of internal state of the cell, biochemical regulation within the cell, and also cell-cell communication. But at the same time, how the cells are able to interact with the matrix, and how the cells are able to sense the physical, biochemical, mechanical signals from the matrix, how the cells are able to change these cues, the physical, biochemical, and mechanical cues in the matrix, and how these are all integrated together to cell phenotype. So with that, I'll actually end the presentation, and I will thank the people that have contributed to my work. So people in Eindhoven, people in Singapore MIT Alliance who have been working with me on the microbiology, as well as the people in National University of Singapore, but also the people in the Microrheology Institute, especially on the cancer metastasis studies. And I will thank also the funding agencies that have provided the financial support for my work. And so thanks all for listening. And I think we have a bit of a time now for question and answer. So feel free to send in your questions, and I'll be happy to answer it.
Thank you, Dr. Kurniawan, for that informative presentation. It is time for Q&A. If you have a question you'd like to ask our speaker, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, if cells have trouble in migration in soft substrate, how can invasion assays in hydrogels and other soft matrices be compared to physiological invasion?
Okay, so let me repeat. Yes, please.
If cells have trouble in migration in soft substrate, how can invasion assays in hydrogels and other soft matrices be compared to physiological invasion?
So I'm not sure I understand the question. So the cells do not typically have trouble migrating in soft substrates. So I think, yeah. So probably this person is asking about the 2D assay that I described in the very beginning, where the cells are able to migrate from the soft to stiff substrate, but not the other way around. So that actually doesn't mean that the cells are not able to migrate on soft substrate. So they're able to migrate, yeah, just fine, but they have some preference to migrate, to be, well, not just migrate, but to adhere on and be on stiffer substrates. But to directly answer this question, so in our assays, the experimental assays are actually in 3D, which are, it's a very different environment compared to 2D. So the cells are actually in the matrix. It is probably the stiffness of the matrix in this 3D environment, the 3D matrix, is not going to be directly comparable to the stiffness or the stiffness readout that you get from 2D substrates. So it's on the order of usually between on the order of 10 and 100 pascals in terms of shear and Young's moduli. And the cells do not have any trouble migrating in this kind of matrices. So, in fact, if you compare this kind of stiffness to the stiffness of tissues in human body in physiology, then it's comparable, for example, to tissues in breast tissues, both in terms of microstructure and mechanical properties. So I hope I've answered this question.
How do you create inner and outer gels technically? Do you have a special 3D chamber or?
Oh, right, yeah. So, yeah, so as I said before, for more technical information on how the assay is done and set up, you can obviously look into our Journal of Visualized Experiment paper, where there is actually a video, a very, very informative video where this is done and how it is set up. But basically what we did was we create a kind of a dome-shaped inner gel. So what I drew as a diagram, it was kind of a little bit deceiving. So it's not really cylindrical kind of shaped gel, inner gel, but it's kind of a dome-shaped gel. And the entire inner gel with the cells inside, they're sort of encapsulated in, well, basically we added the second layer of gel, the outer gel on top of it, and surrounding it. So in practice, it's very easy. It's just a matter of putting one gel on top of or surrounding each other.
Okay. And for this next one, I don't know the name of the journal, so I'll just say it. Which cells did you use in your 2015 JVEXB publication?
Right, yeah. So far we've been focusing on cancer cells. So we've been looking at, breast cancer cells like MDMB8231, but also cells like MCF10A, MCF7, this kind of cell types.
Does the migration through collagen gels change between cells?
Can you repeat the question?
Sure. Does the migration through collagen gels change between cells?
Oh, right. Yeah. So, of course, different cell types have different migration characteristics. Some cells are just migrating faster than others. Some cells are just, have a tendency to sort of walk or migrate slowly. But qualitatively, we are seeing sort of similar behavior. So what I described before in terms of, the cells at the migration front and the cells behind them, and how the characteristics of the migration of these two types of cells are very different, we see them in most of the cell lines that we have been studying.
Have you tried to generate these hydrogels in a high throughput for high throughput screening?
That's a great question. So the answer is no. So yeah, probably there's something that we have to look more into in the future.
Is it possible, one second, is it possible to do the 3D culture in a high throughput way, for example, to use in the small molecule screening or siRNA?
In all likelihood, yes. So far we've been, the experimental assay that we have been working on is kind of not a very high throughput in terms of the matrix. So in terms of the cell, it's very high throughput because in one experiment or in one assay, we can monitor the migration behavior of many hundreds of cells in one experiment. But in terms of looking at not only cells with siRNA interference, but also cells, different types of cells and different disease states, for example, it hasn't been high throughput. So we only have a few assays for studying one cell types, and you probably need multiple of this series of experiments to do that. But in the future, I can foresee that this kind of assay can be made high throughput by miniaturizing. So you probably don't have to create inner and outer gels with the dimensions that we've been working with so far. So we've been working with on the order of like 17 or 18 millimeter gels. Probably it's not necessary to go that big. So in principle, this can be made smaller and in a way that allows you to look at multiple cell types and states of the cells in a more robust and high throughput way.
How do you analyze this data?
Okay, so I'm not sure which data your the questioner is asking about.
We did not specify. This is
yeah. So let's move to the next question.
Yeah. What is the actual method by which you measure this microrheology? Is it a computer monitored process of particle Brownian motion? Can this be used for 2D applications? Would you like to answer those separately, just one at a time? What is the actual method by which you measure the microrheology?
Right. I can try to answer this very quickly. So, yeah. So what we did was we basically looked. We took videos of bead movement in this kind of matrices, and the analysis of the bead movement is done offline. So it's not really an online measurement. So you do the offline measurement by looking at the trajectory of these beads and using generalized Stokes-Einstein equation, you can back calculate or not back out the storage and loss moduli. For 2D application, it is possible. Of course, you have to worry about, for example, if you put the bead on a surface, for example, then probably the motion is going to be affected by things like friction with the surface and things like that. But otherwise, if the environment is isotropic, so that means that there is no difference between the motion of the bead in X, Y, or Z directions, then just looking at the 2D movement of the particles can already tell you something about the 3D local properties.
Okay. Which microscopy setup did you use for imaging the cell migration from inner gel to outer gel?
Right. So we used a confocal setup with equipped with cell culture chamber. So what we did was we looked at some what we call volumes of interest within the outer gel, and we look at several volumes of interest. And we look at we took 3D images as a function of time of these volumes of interest, and by looking at the cells and tracking the cells within these volumes of interest, and in this particular case we are using Imaris, the Imaris software, we were able to track the movement of the cells for a long period of time. So we did up to a couple of weeks of culture.
How do cells absorb drugs in 3D gel matrix? Are the drugs incubated in the gels?
That's right. So we basically put the drugs dissolve in the medium, and we put the mediums on top of the gel. So the pore size of the gels are large enough, you see on the order of 1 to 10 microns, that diffusion of small molecule drugs within the matrix is very, very easy and very, 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 very fast.
Have you examined migration in co-cultures?
That's a great question, and that is again something that we need to look into. But so far, we haven't done that.
What is the current direction on the reason or ideas, or on how the cells feel? Let me start that one again. What is the current direction on the reason or ideas on how the cells feel in their environment, how it translates this physical environment to a chemical signal?
Yeah, so of course, this is a another great question, and this impinges on a very big field called mechanotransduction. So, of course, whatever the cell is feeling on the matrix on the local environment is going to be translated within the matrix, within the cell, to biochemical signals, which then in turn is, influencing the decision making of the cell itself. And so in this presentation, I didn't have time to go into the details of how mechanotransduction happens, but there's a whole lot of literature on that. So I'm pretty sure that it's going to be very easy to find some, for example, review articles on mechanotransduction and just some, a picture of how cells are able to translate mechanical or physical signals from the environment to biochemical signals within the cells. And so the current direction or ideas is, so this is a very big field right now, an emerging field, and people are still trying to understand what is going on.
Is measurement on individual cells or cell population?
So we study the migration of individual cells within a population. So it's not really, cell colony. So if you actually quantify the sort of the average distance between cells in the population is on the order of 5 cell diameters. So there's not really a lot of cell-cell contact going on. So we are really looking at individual cell migrations, but within a population.
Is there a minimal number of cells that can be quantified?
Well, it really depends on what you're trying to quantify, what you're trying to study. So I would say that it really depends on, for example, things like cell seating density. What is the cell seating density that you want to put in and what you want to achieve in the end? So you can, of course, do the same kind of assay where there's a lot of cell-cell contact, for example, by just creating a spheroid or a colony of cells and looking at, things like how cells are able to escape from this colony and migrate into the environment. But at the same time, by going for a very low cell seating density, we've gone, I think, as low as about 50,000 cells per ml, if I remember correctly, and that works fine. So in that case, you will be seeing really, individual cell migrations.
We are almost out of time, so this will have to be our last question. What would be the difference in actin dynamics between 2D and 3D migration?
Right. So that is also an excellent question. So, how cells are able to migrate on 2D has been very, very well documented. There's a review, for example, by Doug Laufenberger and Alan Horwitz. And, how cells migrate on 2D surfaces, the kind of actin dynamics on 2D surfaces is very, very different from the cell dynamics on 3D. And in fact, this is something that we are currently still looking at. And so one of the things that we are looking at right now is what is the role of stress fibers on 2D, 2.5D, and 3D environments. So what I mean by 2.5D environment is structures on substrates that resembles 3D environment, that resembles fiber structures. So we are still looking into this, what kind of contractility, how cells are able to achieve contractility, what kind of forces they are able to exert on the environment, whether stress fibers are as important in 3D as it is in 2D, what kind of focal adhesions are important in this kind of situations, and so on and so forth. So this is really something that is still, being actively studied right now.
I would like to once again thank our speaker for his presentation. Do you have any final comments, Dr. Kurniawan?
Well, I would just like to thank everyone for listening in this presentation. I would again thank the people from Bitplane and LabRoots for organizing this event. And so feel free to just send us more questions if you still have some questions, and I'll try to reply to these questions as soon as I can. Thank you.
Thank you once again. I would also like to thank our sponsor, Bitplane, for making today's educational webcast possible. Just a reminder, if you wish to obtain the credits for today's session, please click on the Get Your Free CME CE Credits button located in the lower left of your screen. This will take you through the process to obtain your certification. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing on www.bitplane.com/search. You will receive an email from Bitplane letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.